Hey guys, this is a lecture related to explanation about burette. I have given you two lectures before this burette related to the apparatus discussion. We are done with the apparatus discussion actually. So this burette was left only, and in this burette you people should know this thing. It is an apparatus which has a controlling tap at the bottom. We can open that tab, we can close that tab just like water dispenser. You can see this thing that in this diagram I have deliberately taken the diagram in which the top part of the burette sorry, and the bottom part of the burette is clear. Okay, so the top part of the burette is telling it to you that you people can fill this burette with the help of a filter funnel. It has a very narrow opening at the top. We cannot fill it as it is directly from the beaker or directly from the flask, etc. We have to use and we should use a filter funnel so that its narrow opening can be adjusted regarding filling of the liquid inside that. Okay, it is very clear that it is a burette which has an ability, a capacity to fill in the liquid to 25 centimeter cube. Guys, I'm going to zoom this. You people can see this thing that it is telling you where is 23.1, 23.2, 23.3, 23.4, 23.5, etc. So it is telling you that it is very, very accurate apparatus and it can measure the volume of the liquid very appropriately. So we can turn on the tap, we can turn off the tap as well. So when we will turn it on, obviously liquid will start coming out of it, and when we are going to turn it off, the liquid will be stopped. So for example, the water liquid is at 10.1. It is just telling you that 10.1 of the liquid is used out of it and the remaining liquid is there. It is just like water dispenser, so it is we are going to use it just like water dispenser. So when the water dispenser is filled, it is telling you that no water is taken out of it, and when we are pressing that button and water is coming out of it, it is just telling you that this much amount of the water has been taken out of that water dispenser. Okay, so I am telling you on the next page. Okay, this is the titration reaction. This is the titration reaction. And what is the titration reaction actually? A titration reaction is basically a reaction between the two components. One of the components I have mentioned it in the previous video. One of the component will be taken in the burette, whereas the other component of the titration reaction will be taken into the yes, whereas the other component of the reaction will be taken into the pipette. I have shown you the pipette in the last video. The partner which is taken into the pipette will be taken into this conical flask. The partner which is taken into the conical flask is from the pipette I have just mentioned, and it will be only 25 centimeter cube always. And you can see this thing that the burette opening is there. We are turning it on and the color changes are observed in this conical flask. There is a stand over there in which this burette is adjusted. Okay, this is a diagram which can tell you this thing more appropriately and clearly. So this is the initial reading. It means that it is the reading at the start, and there is a final reading as well after the reaction in the titration. This is the final reading in the burette. Okay, so burette is there, burette clamp is telling you, and there is a stop clock. We stop cork over there. We can stop, we can open it, and there is a stand. It is very, very clear. Flask is there. Okay, and one of the partner of the titration is in the flask, and which is called as analate. Or you may not memorize this term of analate or titrant, but remember this thing that one of the partner of the titration is taken in the burette, whereas the other partner of the titration is taken in the conical flask. Reaction between the two is happening in the conical flask, and we can observe the changes over there in the conical flask. Okay, so related to this, let me tell it to you again that a titration is basically a chemical reaction between the two partners, between the two reactants. One of the partner is taken in the burette, whereas the other partner is taken in the conical flask. The partner which will be taken into the conical flask is taken with the help of a pipette, whereas the other partner is in the burette, and the burette is filled with the help of a filter funnel. That's all for the chapter number 2. We are done with kinetic particle theory. We are done with the lab apparatus. We are done with the collection of gases and identification test for the gases as well. The next chapter, which is called as purification techniques and separation techniques, that will be our next lecture, inshallah. All the best to all of you. Thank you very much. Take care.